I'm going to describe the new genetic technologies and approaches that are being used in 4 new projects for understanding and improving Atlantic salmon resistance to sea lice. You might be thinking, I'm tired, it's the end of the session, there have been so many promised solutions in the past to sea lice resistance, I don't need to sit through another talk about genetics. But don't press the leave button. These 4 projects are going to have important implications for your future business, and there are going to be some important decisions you will need to make as a consequence of this research. Sea lice, as you know, have a large cost. They cause fish welfare problems. There are no existing control measures that are completely effective, but there is genetic variation in resistance amongst Atlantic salmon, and there are large differences in the resistance of different species of salmon. So the 4 new projects that are starting this year are Crisp Resist and No Lice, which are led by myself with Norfema and funded by FHF and the Norwegian Research Council. Lice Resist, led by Sigbjørn Lien at NNBU, funded by the Research Council, and a new project that will start later this year, led by Ross Houston of the Roslin Institute in Scotland, funded by BBSRC. So first, I'd like to play a short animation describing the Crisp Resist project.
Aquaculture is an important industry in Norway, with millions of Atlantic salmon farmed along the coast, generating millions of meals worldwide each day. But lurking in that sparkling seawater is a challenge: salmon lice. Lice feed on mucus, skin and blood, and if not controlled, can wound and stress the salmon. Lice reproduce and replicate on farms, which exacerbates the problem. Atlantic salmon are particularly vulnerable, but some other salmon species, such as coho, have natural defences against lice. So, what's their secret to success? Coho and pink salmon may not secrete the sort of natural chemicals that attract lice, and immune cells in the skin and blood play a role in responding to lice when they do attach. Changes in gene activity within cells in response to infection at the site of lice attachment can be measured to find important signals driving the salmon's immune response and investigate the ability of lice to suppress the host's immune response. Gene editing is a powerful new technology with high precision. It will allow us to test gene functions and assess possibilities for mimicking the lice resistance mechanisms found in other salmon species. For example, it could be used to prevent production of lice-attracting molecules, hiding or disguising the Atlantic salmon from the louse. Like testing a vaccine, we can test the effectiveness of editing particular genes in a biosecure facility, and model how these technologies could be most effectively used to immunise the entire farmed population of fish. The Crisp Resist project will fully explore the potential for using gene editing to prevent lice infestation. This will allow society to make informed decisions about whether and how these technologies should be deployed.
The motivation for these 4 projects are to find the genes that are affecting lice resistance, which will give us the ability to test if gene editing could be used and if feed additives might be effective. To increase the breeding accuracy, which will increase the rate of improvement in resistance, and to devise strategies for most effectively and responsibly implementing the technologies. So these 4 projects we hope will provide breakthrough solutions for the industry. So first I want to highlight some of the new technologies we're using to identify genes involved. First, we'll identify compounds secreted from the salmon skin with quantifiable differences between high and low resistant fish using mass spectrometry. We'll isolate these compounds and we'll test them on salmon lice, measuring the swimming speed, hop frequency, and the distance travelled by the lice towards the source. And this will reveal lice attractants that might be masked or eliminated to boost resistance. There are two technologies that will allow us to study skin gene activity in space and time and cell type after infection. So here on the left down the bottom is a section through a salmon louse, SL, attached to a gill filament. We'll be using spatial transcriptomics to reveal gene activity at the attachment site of the lice, which are represented by those glowing red dots, and single nuclei RNA-seq to reveal activity of genes in specific cell types at the attachment site over time, which are represented on the right-hand side by the glowing cell type. So this will allow us to identify genes responsible for host resistance, success, or failure. We'll also be using proteomics to study how the louse is able to suppress the immune system of the salmon host, and we'll look for ways to break this mechanism of immune suppression. The activity of specific genes is locked under some conditions, and it's unlocked under other conditions. NMBU will use the latest new sequencing technologies to look at what genes are locked and unlocked away from and at the site of attachment in the different species of salmon. This will give us another approach for studying gene activity associated with resistance. Once we have knowledge about the genes and mechanisms involved, we need to determine how we can use this information to boost the ability of Atlantic salmon to resist lice. So we'll test the feasibility of 3 different strategies for boosting the resistance of salmon to lice. Gene editing is a Nobel-winning prize targeted way to knock out or upregulate genes we are interested in. It uses CRISPR-Cas9 molecular scissors, and we will check for desired changes in gene activity and side effects, and we'll compare the lice resistance in edited fish to control fish to test gene function and to look at the potential to boost resistance by the host to the lice. Once we have edits we know boost resistance, we need to determine effective and responsible ways that gene editing could be implemented and disseminated. To do this, we will develop simulation models and we will work closely with industry partners. Finally, we will design feed additives based on knowledge about the gene pathways and mechanisms discovered in the project to test the feasibility of using feeds to mask or repel the lice from Atlantic salmon. So the project will deliver new knowledge about the genes behind these resistance mechanisms, potential of gene editing, feed additives, and genomic selection. It'll give us strategies for slowing reproduction and spread of the parasite, for the creation and dissemination of edited fish, for the application of genomic selection, and for minimising adaptive response by lice and mitigating other risks. So the industry will be armed with the knowledge it needs to solve this problem. So in 4 years' time, you will face a number of important decisions. Likely, we will have a much greater understanding of natural processes affecting resistance to sea lice and strategies that could transform the Atlantic salmon industry as we know it. These projects will advise on the most responsible and effective approach or combination of approaches. There's going to be a need for further testing of gene editing to ensure there are no risks. But together with the industry, we need to have an open dialogue and to build public trust around the use of these technologies. So I want you to ask yourselves now, is business as usual with no change an option for you, considering the effects of sea lice on profitability, welfare, and public perceptions? Our goal in these 4 projects is to develop safe and effective strategies, but you will need to work with the public and government agencies to decide if these strategies should be implemented and to clear the pathway so that you can solve this problem and transform your business. Thank you.